Good afternoon or good morning, depending on what area of the country you're joining us from. I'm Milo Azad. I'm one of the ICRN members, the vice chair, and I'm very happy to welcome Dr. Gores here today for our seminar series. Today, Dr. Gores is going to be speaking about a really important topic, especially for those of us who are interested in immunotherapy-based approaches and are really trying to understand the animal model systems that are present for cholangiocarcinoma. So he's going to today talk about the advantages and disadvantages and challenges of the animal model systems that exist in cholangiocarcinoma. Dr. Gores comes to us from Mayo Clinic in Rochester, Minnesota. He's a lab-based researcher that has really been focusing on liver and liver-based cancer diseases, really trying to understand both predisposition to the development of malignancy and then how both transformation happens, metastasis happens, and how we might target these using important model systems. He presently serves as the executive dean for research at the Mayo Clinic, as well as the Eisenberg Endowed Professor in gastrology and hepatology, and we're very lucky to be able to learn from him today. So welcome and thank you very much.
Thank you, Neil, for the nice and generous introduction. So it's my pleasure to be speaking about animal models for cholangiocarcinoma for the ICRN in conjunction with the Cholangiocarcinoma Foundation. So I just want to highlight that John, who drove around the country highlighting awareness for cholangiocarcinoma, was in Rochester here with us, and I just want to highlight some of my colleagues who also have an interest in this disease, Lewis Roberts and Samara Ilias. And we had the honor of taking one of our more distinctive buildings on campus and putting a green light on it in honor of the foundation and its efforts. So I'll be speaking about the animal models of cholangiocarcinoma. I think for those who would like to read on this, I think this paper in Cancers a year ago is really a wonderful overview of it, and I'll be using some figures from this paper. So I'm supposed to talk about the advantages and disadvantages and compare and contrast models, and I'll do that along the way. So if one sets out and says, what's the ideal animal model for cholangiocarcinoma? It would mimic an anatomic subtype of cholangiocarcinoma. It would be based on human genetics. It would duplicate the tumor immune microenvironment of human cholangiocarcinoma. It would duplicate the pathogenesis of tumor genesis for this disease. It would be efficient in time and cost and resources. So we're not there yet, but we're seldom in the ideal state, but this is what the field needs to achieve over time. So this is a figure from the paper I mentioned, and this will talk a little bit about the evolution. The first model of cholangiocarcinoma we really had was the Furan model developed by Al Sirica at Virginia Commonwealth University, and it was in rats. And they developed the intrahepatic cholangiocarcinoma. And I'll come back to that. Others have used thioacetamide in rats, and really diethylnitrosamine and dimethylnitrosamine more likely to induce hepatocellular carcinoma than cholangiocarcinoma, although sometimes one can get mixed tumors in the rats. So these chemically induced animal models have been primarily applied in rats and have not been widely utilized. The transgenic models, which are a type of genetically engineered mouse model, where one introduces mutations into the germline of mice and then crosses them with other mice, so one might get an animal model that's deficient in the tumor suppressor p53 and cross it with an animal that has an activated mutation of KRAS, for example, is one of the published models of transgenic mouse models. They primarily produce tumors in hepatocytes, So this is hepatocyte-derived cholangiocarcinomas. We don't have real good specific promoters for intrahepatic cholangiocytes. There's been a lot of work done with that. So these have been primarily hepatocyte-driven cholangiocarcinomas, and we have to keep that in mind as a potential defect. These are very time-intensive and labor-intensive in terms of breeding mice and then waiting for offspring, doing genotyping the mice, and then waiting for them to develop tumors, and the tumor penetrance will never be 100%. Another approach is to use DNA or RNA and do hydrodynamic injection into mice. We'll look at this a little bit more detail later. But these induces genes preferentially in hepatocytes in the liver, and then one can obtain liver cancers, shortcutting the breeding necessary for the transgenic mouse models. Again, this has the deficiency of that the cholangiocarcinomas are intrahepatic and they're derived from hepatocytes. But this one is widely used. People, of course, have used a primary human cholangiocarcinoma cell lines or spheroids, and now people are moving more towards organoids or even patient-derived tissue, often to the flank of immunocompromised mice, and these would be the subcutaneous xenografts or patient-derived xenografts. You can also, the problem with these is that they don't recapitulate the tumor immune microenvironment, so you have a species mismatch, as you have a mouse, a stroma, and then a human cell line. So it has some deficiencies. Second, they're often not placed orthotopically, but rather subcutaneously for ease of monitoring growth and surgical approaches. But again, this also fails to recapitulate the native environment of the liver. For example, the liver is very rich in a macrophage population termed Kupffer cells, and has a dual blood supply, is rich in pericytes termed stellate cells, which can differentiate into myofibroblasts, become cancer-associated fibroblasts, and one loses all that sort of cell-cell crosstalk and species matching in this model. And more recently, people have started to use spheroids or organoids and inject these into orthotopically into mice. And we can explore this in a little bit of detail also. But oftentimes, when one's taking human tissue, again, one will have the species mismatch. Okay. So the ideal animal model for cholangiocarcinoma would mimic an anatomic subtype. And although there's been a lot of miscoding of cholangiocarcinoma, there are really three anatomic subtypes: intrahepatic, the perihilar cholangiocarcinoma, and the distal cholangiocarcinoma. And in all the animal models to date have been intrahepatic cholangiocarcinoma. But we recently have a paper that was just published that I'll review online, which is the first model, if you will, of extrahepatic cholangiocarcinoma. So a recent advance, but most of the models are intrahepatic. And why is this important? Because these are really different types of cancers in terms of the cells of origin, and the behavior of the cells. So the intrahepatic cholangiocarcinomas presumably are derived either from hepatocytes through transdifferentiation and metaplastic processes, but more likely are derived from the small bile ducts or the interlobular bile ducts, either small cuboidal cholangiocytes. And in conventional intrahepatic cholangiocarcinoma, they're either very extremely desmoplastic, as illustrated in the far left slide here, or they have a lot of cuboidal epithelial cell processes with glands, as demonstrated in this small duct cholangiocarcinoma. Whereas the perihilar cholangiocarcinomas may arise from either large bile duct cholangiocytes or even the Tumor-Derived biliary glands, so a different pathogenesis here. And they're often produce a lot of mucin, mucoid-producing tumors along with the desmoplasia, and they tend to have a larger cell type. So by histopathology, by the cell of origin, the anatomic location is important and produces different anatomic or pathologic subtypes of cholangiocarcinoma. So I mentioned that when I was reviewing animal models, they were virtually all the toxin-induced and gene-induced and hydrodynamic approaches. The cell of origin is a hepatocyte, and they form intrahepatic cholangiocarcinoma. So some concern with those models. The first extrahepatic biliary cancer in mice was just is published ahead of time in Cancer Discovery. It comes from a German group, and they used the promoter PDX1 pre-expression. So PDX is important in extrahepatic biliary tree, pancreas, and duodenal development embryologically. And they found that suppression of a cyclin kinase inhibitor, so permitting the activation of cyclin kinases, plus a PI3 kinase activation induces extrahepatic biliary tract cancer. This is their work. They emphasized that embryologically intrahepatic bile ducts and extrahepatic bile ducts have arrived from different mechanisms embryologically. And that, as I said, the extrahepatic biliary tree, gallbladder, duodenum, and pancreas are driven by a PDX positive approach. When they crossed these animals initially in the rosa locus and activated PI3 kinase, they were able to introduce sort of intraepithelial neoplasia in the extrahepatic cholangiocyte compartment and also in the pancreatic ducts. And these were the proportion of patients. And then very interestingly, they went back then and they said, okay, what if we do this with a KRAS activating mutation? As we all know, KRAS is very important in pancreatic cancer. They had a great animal model using this PDX driver of pancreatic cancer, but they got no cancers in extrahepatic biliary tree. And so this is a really striking example of tissue specificity of carcinogenesis. So we have the same embryologic origin of the tissue. We have a well demonstrated that this promoter worked in the duodenum, pancreas, and extrahepatic biliary tree and gallbladder. And then KRAS only produced pancreatic cancer and did not produce extrahepatic cancer. So a very striking example that there are changes between ductal cells in various tissues and tissue specificity that really drives carcinogenesis. They were able to subsequently work this out through a very sophisticated approach, which was unbiased approach to look for genes which may drive carcinogenesis in the biliary tree. And as I said, this is a cyclin kinase inhibitor. And by decreasing this, they got extrahepatic cholangiocarcinoma, invasive. In contrast, they were able to duplicate what is well known, that you take the activating mutation of KRAS at the G12D site and lose P53, you'll get pancreatic cancer. So interesting biology of carcinogenesis in the upper GI tract, but more important, our first model of extrahepatic cholangiocarcinoma. And I think this will need to be exploited as we move forward. Now, as I said, we have to remind ourselves that these cancers should be based on the human genetics of cholangiocarcinoma. And I want to highlight first that about 15% of patients with cholangiocarcinoma have germline mutations. And these are the sites. This was on data that comes from Mayo Clinic, Arizona, primarily by an unbiased approach, just taking patients coming in with malignancies and looking for germline mutations, and when this was very frequent cholangiocarcinoma. So this has to be revisited in great detail. And then we're all aware of the intrahepatic cholangiocarcinoma somatic mutations in isocitrate dehydrogenase, BAP1, ARID1A, B, the FGFR2 fusions, and then absence of the loss of P53, PI3 kinase pathways. And so intrahepatic cholangiocarcinomas to be have fidelity to the human condition to really be driven by these tumor alterations. And the extrahepatic biliary tree is different. So there is KRAS in extrahepatic biliary tree. So the animal model is interesting that they could not recapitulate that in mice. You have P53, but then you also have ARID1, but some other receptor tyrosine kinases come up, such as EphA2, and then a variety of often epigenetic modifiers. In contrast, isocitrate dehydrogenase mutations and FGFR genetic aberrations are very unusual in the extrahepatic biliary tree. And then this would be the sort of the structural landscape of extrahepatic biliary tree, again highlighting these pathways, particularly the epigenetic modifiers, which I think have not been exploited yet in animal models of cholangiocarcinoma, remain quite important. Okay, so taking into context the different anatomic subtypes, their genesis of those tumors, and their differences in genetic aberrations, we can come back and then explore these animal models that we talked about. I want to highlight the use of, go back then I'll talk about the cholangiocarcinoma cell lines and the organoids and patient-derived tumors. So these are time-honored approaches of placing these into nude mice in a subcutaneous approach. The strength is the reproducibility. It's technically facile, and you can monitor tumor progression quite easily. On the other hand, as we mentioned, there's the absence of the tumor microenvironment. This is an immunodeficient host with often the tumor and the host being mismatched in terms of species, and therefore the microenvironment is simply wrong. Okay. There's been a lot of interest in patient-derived xenografts for cholangiocarcinoma. These are usually intrahepatic because the tumors are more easily identifiable, and there's excess tissue that the pathologist doesn't need, and so one can take these. We've been quite proficient. We have about 30 or so different type of cholangiocarcinoma xenografts in the institution. So the tumor can be harvested. Small pieces can then be obtained, and they can be implanted into the animals, and they will grow over time. And then they can be propagated, they can be frozen and used repeatedly. There's a big paper in Nature Communications a week or two ago that looked at the fidelity of various xenografts to the parent tumor. They maintain genetic fidelity. There's not a lot of genetic drift over time. They were, of course, pure in the genetics of the parent cancer, mainly because they had lost the tumor microenvironment. So they used that as an advantage in the paper, but one could equally perceive that as a disadvantage. You can also obtain them from biopsies. So you can, with a second pass or third pass, you can get sufficient tissue and implant it and then you can get a cholangiocarcinoma. As you see in this histopathology here, there's very little stroma present. Okay. The advantages people have used these for has been classic testing of anti-cancer agents. For example, here you see gemcitabine and oxaliplatin being tested in xenografts. And you can see the control tumors have the tumors, and then those animals who have been treated may have a response. So in some ways, they can recapitulate the human condition despite the absence of the tumor microenvironment when one's looking at cytotoxic therapy. This is an interesting model that sort of uses a grafting approach, which I found intriguing. Others may find contrived. So the idea was you would take liver progenitor cells, generate organoids in culture. You would then transduce the cells with oncogenes, and then you could re-implant them into the Syngeneic model and study cholangiocarcinoma. So you bypass all the germline mutations in making transgenic animals, with understanding that that's somewhat simpler these days in CRISPR-Cas9 world and gene editing. But still, there's still a lot of breeding involved. So this is a shortcut to that. So in this paper, they took these liver progenitor cells and got classic organoids. As I said, they did genetic manipulation in vitro, and then put them back into the tumor, and they got into the animal orthotopically, and they got tumors. And the tumors look like cholangiocarcinoma. And what this paper did, which is quite fascinating, is they put in an FGFR2 fusion into the organoids at this step. And then they were able to generate the fusion model of cholangiocarcinoma in mice. To my knowledge, nobody's been able to do that with a germline approach. In other words, if you make a transgene for FGFR2 fusion, I'm not aware of a publication showing that you actually get cholangiocarcinoma. If you do it in the progenitor cells in vitro and then put them in situ, you can get tumors. And then they were able to look at bypass mechanisms for tumor growth. So they inhibited FGFR2 fusions with BGJ398, which is now approved for treatment of human cholangiocarcinoma, and showing that they were escape pathways or resistance pathways, and then they could put in a MEK kinase inhibitor and show that this MEK kinase pathway was a bypass pathway or a resistance mechanism or a compensatory pathway, and then the combination of the MEK inhibitor with the FGFR2 inhibitor was salutary. So this is an interesting animal model combining both organoid technology, gene manipulation in vitro, and then carcinogenesis. A little bit on the hydrodynamic in transfection for inducing liver cancers in mice. This often uses a gene under a very strong promoter, such as EF1 alpha, and then using the Sleeping Beauty transposase. So this is in a transposon. This is in a transposase. One injects these very rapidly in a large volume of saline for the mouse. Injected via the tail vein in about 7 seconds, realizing the mouse volume is about 5 milliliters of circulating blood. So this is a significant volume overload. Somehow, the liver, as it absorbs this excess fluid, the hepatocytes become sensitive to spontaneous transduction by a physical process. And these oncogenes then become, by the transposon transposase mechanism, become stably expressed in human mouse hepatocytes. And one can get models of cholangiocarcinoma using this approach, and Xin Chen and Diego Calvisi have popularized this approach for both generating HCC, but also for cholangiocarcinoma. As I said, I'm not so sure they duplicate the pathogenesis of cholangiocarcinoma, and that the tumors originate from hepatocytes. But as I said, because of its technical ease and efficiency and reproducibility, many investigators use this model. Okay. Now, Neil mentioned the interest in the tumor immune microenvironment, and so I'll take you a little bit on on some work that we've done. So we collaborated with Xin Chen at UCSF and thinking, well, why don't we use the transposon and the Sleeping Beauty transposase approach, but why don't we directly transduce the biliary tree? And so the idea that we could put in the genes of interest, and then I'll go over this in detail, but you can transduce the biliary tree and get tumors. Interestingly, these tumors are largely intrahepatic cancers, and presumably they easily transduce the intrahepatic biliary tree and metastasize intrahepatically. With all full disclosure, my goal was to develop an extrahepatic biliary tract model here, but I got an intrahepatic model. So what we do is we clamp the distal duct gently, and then we distend the biliary tree in the animal by entering the gallbladder with the volume. We then want retention, but also we want to induce some inflammation to promote carcinogenesis. So we tie off the duct to the left lobe of the liver. We do a cholecystectomy so we won't get a gallbladder leak, and then we remove the clamp. And we give IL-33 post-op for about 3 days. And this was, again, based on the concept that IL-33 was a mitogen for the biliary tree. And then I thought that by acting as a mitogen, it would open up the chromatin and allow more efficient transduction of the biliary epithelia. One could postulate it's also a damp and produces inflammatory response. It's an inflammatory response that promotes carcinogenesis. Either concept is probably valid and probably both operational. And then over time, we get these tumors. The H&E, they look like cholangiocarcinoma. They have the transgenes. We've used myristylated and therefore constitutively active AKT, this phospho-AKT, and phospho-AKT is quite usually present in human cholangiocarcinoma as well. And then we've used a constitutively active YAP, which is the effector of the Hippo pathway, and a co-activating molecule in transcription, quite potent and present in about 60 to 80% of cholangiocarcinomas. We showed that they were indeed phenotypically cholangiocarcinomas and that they were stained positively for cytokeratin. They stained negative for a marker for hepatocytes, which is HepAR1, and they also had expressed SOX9, which is also a marker for cholangiocarcinoma. And they recapitulated the tumor immune microenvironment. So this is what we were most happy with. Cholangiocarcinomas are desmoplastic. As we mentioned, the liver is rich in stellate cells, which when become activated, express alpha smooth muscle actin, like all myofibroblasts do. They do change they're a little different from classic myofibroblasts, which are activated in cirrhosis, in that they express periostin and tenacin C, which cirrhotic activated myofibroblasts do not. So these have been educated by the tumors. And there's a lot of neoangiogenesis as demonstrated by CD34 staining. We have established cell lines from these, and Xin Chen has now, where I work from her laboratory, was she's established organoids from the hydrodynamic model. These cells, we established several cell lines, but we preferentially use something called SB1 because it grew the easiest. But all the cell lines express SOX9, CK7, CK19. They did not express HNF4 alpha, which is a marker for hepatocytes, nor did they express markers for myofibroblasts. And again, one could see that they expressed the oncogene YAP, which was flag tagged in transduction. We have transplanted these into syngeneically into black 6 mice, and we get tumors. And the tumors are present by 4 weeks, but sometimes 8 weeks. And as I said, they can form a primary lesion, then they metastasize intrahepatically. Dr. Ilias has used this to study the tumor immune microenvironment quite successfully. He continues on those studies. She had a nice paper in JCI last year, and she's speaking in November at this forum, so I won't supersede her discussion of what she's learned studying the tumor immune microenvironment, but this model has proven very effective for that purposes. We've shared it with multiple labs, happy to do so. And including some work that Tim Greeton has published. Tim sequenced of the cells are able to show that they profile with a subset of intrahepatic cholangiocarcinoma as well, based on expression profiling. So these syngeneic models have the strength of recapitulating the tumor immune microenvironment in an immunocompetent host. They have quick tumor development. The weaknesses that they require some abdominal manipulation. There's always a surgical risk. And monitoring tumor progression non-invasively probably can be done, but it won't be easy. One would have to have very superb small animal imaging studies. We did start to do some of the small animal imaging study by ultrasound, and we could certainly see the tumors by ultrasound, but then the question becomes marking a specific tumor and then following its growth, and all the aspects of nuances of monitoring progression and regression of cholangiocarcinoma in human trials by cross-sectional imaging studies are apparent in the mouse. I talked a little bit in the early about whether we should, about mimicking the liver situation. So hepatobiliary inflammation is a risk factor for cholangiocarcinoma. Is the genesis of cholangiocarcinoma arising in that environment different from that which does not arise in the background inflammation? And there's an interesting paper that sought to address this issue by using an animal model of bile duct inflammation and then using a genetically altered mouse. So they used the activating mutation in KRAS, and they did this in a specific in a conditional manner in hepatocytes. Then they used DDC in the diet. So DDC is a porphyrin. It is absorbed, gets excreted into bile, loses its solubility, and then actually precipitates out in bile canaliculi and very small bile ducts. And the precipitation, if you will, then is like microlithiasis and is obstructive to the biliary tree. You get a cholestatic pattern that's accompanied by inflammation. So for those of us who study biliary tract disease and its consequences in terms of inflammation and fibrosis, it's probably the easiest model to adapt. And one can have a variety of issues here. They had the transgenic background. They fed the DDC for 2 weeks, and then let the mice recover because the mice lose weight on the DDC diet, and then they pushed forward after the mice had adapted. They pushed forward with DDC over time. One can also use this cycle in an interrupted manner, which is our preferred approach, is 2 weeks on, 1 week off over time to come to a state that's useful for your studies. In validating this model, they were quite sophisticated and they did lineage tracing. They had a model where the hepatocytes were expressing a red fluorescent protein. They could inject by hydrodynamic injection the AAV8 serotype on a TBG promoter with Cree into the animals. That will remove expression of the red fluorescent protein and allow expression of a green fluorescent protein in the hepatocytes. So you can tell by color now what was derived from a hepatocyte and what was derived from a biliary cell. And so you can see that using this approach, the hepatocytes were indeed green, a very high transfection efficiency using the AAV8 serotype with the TBG promoter. And the non-parenchymal cells remain red, including biliary tract cells. And then you can see that there's some ductular proliferation present here in this model. And so they could track the origin of the early stage malignancies in this model. And they were able, with the presence of the intrahepatic KRAS, with inflammation, to induce foci, neoplastic foci. These were not necessarily invasive cancers. They were neoplastic foci, showing that one can drive carcinogenesis with inflammation in the mouse. Efficient in time, where do I think the field is going? I think the field will be using more and more 3D models with implantation. That's where I think things will go. So using either mouse cancer organoids or human cancer organoids, one will be able to hopefully put these into mammalian models. In the mouse, one should be able to use syngeneic transplantation models. With human organoids, one can either use the xenografts, but one can do better orthotopic xenograft implantation. And then one will compare and contrast in vitro observation versus in vivo observations. But as Nilo mentioned, I think that one studying the tumor immune microenvironment, one's going to be using syngeneic transplant models of mice. So what the future animal models of cholangiocarcinoma, we need better definition of anatomic subtypes. I think that the PDX1 Cree model will have to be explored by others. We had actually started in collaboration with John B at Mayo Clinic, Florida, using the PDX1 model. The Germans got there first, but we had made all the observations they had this as an extrahepatic driver for cholangiocarcinoma carcinogenesis, and interesting, it leaves the pancreas alone. We need better fidelity to the human genetics of cholangiocarcinoma and driver mutations, and particularly maybe germline mutations. We need better definition of the tumor immune microenvironment as it relates to human cholangiocarcinoma. I think the issue here is we don't really understand what is the human tumor immune microenvironment. We don't know how many, what macrophage subtypes are there. We don't understand what myeloid-derived suppressor cell types are there, how they crosstalk with the cancer-associated fibroblasts. And so we'll need to continue to work in parallel with our mouse models and the human models to get better insight into that. And Dr. Ilias will speak about that in November. We need better focus on the role of inflammation. So I think that the DDC model provides us an axis there, and is a way to sort of preset the inflammatory background to foster carcinogenesis. And we will, as I mentioned, we'll need greater use of syngeneic transplant models because of their efficiency and their fidelity to the tumor immune microenvironment. So I thank you for spending time with myself and Bile Duc Bob as we've taken a tour of cholangiocarcinoma. And as I like to say, we all need an imaginative friend, an imaginary friend. So Bile Duc Bob is my partner as we explore animal models of cholangiocarcinoma. Thank you.
Thank you so much. That was a whirlwind tour of years of your work and many others' work as well. So I want to thank you again for coming. I have put in the chat box that any questions that people have, please enter into the chat box, and I'll try to moderate any of those questions. But since I'm here, I'm going to go first. So this weekend, I'm actually giving a talk talking about the different classification schemes that exist for cholangiocarcinoma. And there have been many groups that have talked about the way that we may biologically cluster patients, which really steps outside of the anatomical classifications and looks at the underlying biology. And I think arguably the group that has done it the best so far is from the cancer discovery paper from a few years ago, where there were 4 major subclasses that were found when you combine genomics and epigenomics, as well as expression. So I'm just curious how we take that kind of work into prime time, both clinically, but for you and for what your talk is, how do we use that to inform the animal model systems that we're using?
Yeah, that's a really great question. Thank you for the question. So I have to tell you that you know, I've spent a lot of time also thinking about hepatocellular cancer and all the different subtypes that have been defined by looking at genetics. And I would say that from a clinical perspective, and I'll switch back to my clinician side, that none of those subtypes have demonstrated clinical utility in hepatocellular carcinoma. One could argue that is because that disease has so much heterogeneity. And so you have heterogeneity between patients, you have heterogeneity between nodules, you have heterogeneity within a nodule. But having watched a decade of that science play out, having participated in it, reviewed papers in that area, etc., I'm a little skeptical of that approach for subtyping liver cancers. That would be my first comment.
Mhm.
Now, my second comment would be that for the sake of discussion, there's validity to the approach. I think we just need better profiling of the animal models, which we haven't done.
Mhm.
And my guess is that we'll find models that match those subtypes. And first off, I mentioned that Tim had sequenced our cell line and showed that it matched 30% of a subtype of intrahepatic cholangiocarcinoma. So I think the work just has to be done if one wants to match it that way.
Yeah, I actually completely agree about the subclassifications as well. That was my eventual conclusion from going over the literature of the last 10 years, is that in the end, the individual molecular mutations and abnormalities are the thing that have been the most defining in terms of prognosis and therapy, and all of the rest is helping us understand biology, but I think we're still years away from knowing how to really use them. And I think that's probably true preclinically as well. So Greg Lazinski asked a question. Can you comment on whether adding luciferase or other reporters in CCA cell lines elicits immunogenicity in the liver tumors, or is this an approach that we can use to track longitudinal growth in orthotopic models?
I thank Greg for logging in today and listening and appreciate his attendance. So we did put luciferase in, and it really hindered tumor growth. So we came to the conclusion that at least in the black 6 mice, the luciferase was immunogenic. And so we abandoned that approach. We've also observed that sometimes transducing the cells, just simply transducing the cells in vitro and putting them back in can alter their immunogenicity. And so we have had to abandon, which I thought were really promising studies we transduced the cells and knocked out various chemokines, et cetera, to study the role of the cancer cells in the tumor immune microenvironment. And that also hindered growth. And the presumption was that the transduction process itself can alter immunogenicity of the cell. So either by the transduction process and selecting, or as Greg suggested, simply using the luciferase. At least in our hands with our SP1 cells in the black 6 mice, we think that it elicits an immune response in its tumors and its anti-tumor.
It's such a great question, especially for a lot of our trainees that are on the call as well, that now that we have so many different tools to manipulate cell lines, that each of those actually has to be tested separately to make sure that that tool itself isn't causing changes, especially when it comes to immunologically. So yeah,
I was so shocked, since we have a little bit of time here, so we knocked out a chemokine to prevent monocytic monocyte lineage coming into the tumor immune microenvironment, and we phenotype, and then Samara says, well, you got to do the mock transfection, and we had the same effect with the mock transfection. So one can artificially alter the immunogenicity of the cells.
Amanda Ruggieri, I think also from memory, asks, in order to recapitulate the many subtypes of cholangio, will we also see that we have to have several different animal models for cholangiocarcinoma, I'm assuming for different experiments, and how relevant would you consider each model for overall advancing the field?
Yeah, I think it's a great question, and I think we will. I think we'll have to have different models, and at some point, I think we'll have to have different models for intrahepatic. Of course, we'll have to have different models for extrahepatic, but probably as we look at the tumor immune microenvironment, different genetic models will produce different responses. So we have also collaborated with others and gotten other cell lines which have a different genetic aberration. And they have subtle but probably important differences in the tumor immune microenvironment. So, but the human cancer is very heterogeneous too. So at the end of the day, what you'll be studying will be relevant to a subset of human cancer, and you'll have to have multiple models if you want to cover the whole spectrum.
Yeah, I think a related question, several etiologic factors such as HBV, HCV, liver fluke, microbiome have been linked to cholangiocarcinoma. Are there any efforts to incorporate these various etiological factors to new mouse models of cholangio?
Yeah. Yeah, a fair question. One people have thought a lot about. So I will start backwards with her questions. People have tried to use the liver fluke in the mouse, and it doesn't work for whatever reason. It will work in golden Syrian hamsters. The problem, of course, is the genetic background and the reagents for studying disease in the hamster are quite limited. And so the models never really caught on. So the liver fluke doesn't work in the mouse. I will say that we just, if you're interested in liver fluke carcinogenesis and other things, we just had a review in Nature Reviews in Disease Primers come out yesterday or today, with a great deal focused on fluke biology and its role in carcinogenesis. A second, there are no mouse models of hepatitis B or C that are really valid. That from a pragmatic perspective just simply can't be done. People have done a lot with fatty liver and liver carcinogenesis. And that's probably become a more important model of looking at the background of human disease. I would strongly encourage people who are interested in cholangiocarcinoma to consider that model. And I think we have made some forays into that area. We have a very good model of non-alcoholic steatohepatitis. We're quite well versed in that disease process. We haven't found striking phenotypes, but that doesn't mean that using different cell lines or different approaches, one won't. So I would strongly encourage work in that area.
All right. Well, I thank you so much for your time this hour, and I'm sure that we are going to be seeing lots of people logging into YouTube to add to the people that are here as well. But I really appreciate it, and I hope you have a wonderful weekend to you and to everybody on the call.
All right, thank you, Nilo, and thank you for the opportunity, and I thank the organizers for their efforts to bring this to fruition. It's greatly appreciated by the community.
All right, take care, everybody. Bye-bye.
Bye-bye.